Nucleic acids are made of sugar, phosphate, and nitrogen bases, how they come together. So they're double-stranded, okay? And they make this helix, what we say is kind of a right-handed, double-stranded helix that coils around. This is my little Pee-wee when he was like 3 years old, standing in front of a big sculpture of DNA. Bless you. And actually, this is the one of the discoverers of DNA structure and how DNA, the architecture of DNA, James Watson. I don't know if you guys know Watson and Crick. If you've ever heard of Watson, that is Watson. And I made my kids just stand next to him. I'm like, go stand next to him real fast. They had no idea. They're like, okay. He's kind of crabby. I have to say, he's kind of crabby. I mean, he's old. I know everybody gets kind of a free pass. He's like crotchety and crabby. And he's like, oh, God. But he took a picture with us, and he is one of the key contributors to figuring out the structure of DNA, okay? And really what the structure is it's two strands, so it's double-stranded. It's made of a sugar-phosphate alternating backbone on both sides, sugar, phosphate, sugar. So it's a deoxyribose sugar and a phosphate. And they're bonded together, and they alternate all the way down, so they make up the strand on each side. And then you've got the bases, the nitrogenous bases that are in the middle. And those are connected to each of those sugar-phosphate units. And the ATCG, these nitrogenous bases, adenine, thymine, cytosine, guanine, are the alphabet of DNA. This is the lettering or the code, the alphabet of DNA. The order of this in their sequence is what is the code or the blueprint of your DNA for making all of the proteins of your body that we just talked about. Those proteins and their amino acid structures, their structure, that sequence comes from this sequence of the alphabet of DNA. The A, T, and the C, G. Now notice A, oops, sorry, I lost it. A always pairs with T, and C always pairs with G. Have you guys learned about this already? A little bit somewhere, right? Okay, so this is real basics of DNA. We've got nucleotides, which are the individual monomer of the phosphate group, the pentose sugar. That means it's got 5 carbons. It's not a hexose like glucose that has 6. It's a pentose like ribose that has 5 carbons. 1, 2, 3, 4, 5. And it's got a nitrogenous base. It's got a nitrogen-containing base, one of these types right here. That is the nucleotide. When you put that together and we covalently bond it through these linkages, through the phosphate, ribose, phosphate, ribose, phosphate, ribose, that together is a nucleic acid, okay? And notice at the end it has a hydroxyl group. It's got a 5' and a 3' end. So the number 5 carbon sticking off, it's got its phosphate sticking off, and then the number 3 carbon on this one at the bottom has its OH free on the bottom. Again, DNA has polarity. Like proteins, one end is different from the other end. So we say that DNA has a 5' and a 3' and it has polarity. This whole entire thing is the nucleic acid. This is the nucleotide. The nucleotide is then what? The monomer or the polymer? The nucleotide is the monomer. This is the polymer of the nucleic acid. In the final form is the nucleic acid. These are the different nitrogenous bases. If we look at them, cytosine, thymine, uracil, that's only in RNA. We'll look at that in a minute. Adenine and guanine. Okay, so basically it's a polynucleotide. Poly, like a polysaccharide, remember those? Polynucleotide. Lots of nucleotides bonded together makes up the nucleic acid. DNA specifically is called deoxyribonucleic acid because it's a nucleic acid, okay, because it's got the acid part of it, and it's the sugar phosphate backbone. But the sugar is actually the deoxy version of ribose. So ribose is the sugar right here, okay? And normally, so this carbon right here, the number 2 carbon, there's no hydroxyl there, there's no OH. So there's no oxygen there, so we just say that this particular sugar, this ribose sugar, is deoxygenated. So it's deoxyribo for the sugar, nucleic acid. Okay, so that's where DNA, that's where the term DNA comes from. Deoxyribonucleic acid. The ribose sugar just lacks the oxygen at the 2' position. This is a lot of fancy chemistry that went into this in biology and work. To take a crystal, turn DNA into a crystal, send x-rays in it, look at the diffraction patterns, put a map together, and then a model together based on its overall structure. And this is an image taken from some of these actual diffraction and X-ray crystallography diffraction studies that were done. And even though everybody emphasizes Watson and James and Watson, Crick, for identifying the structure of DNA. Really, you guys have probably heard of this too, Rosalind Franklin, she was a grad student actually at the time, and she was the one who actually did the actual photos and images of these. That allowed the other guys and her to put together the overall structure and go, oh, it's a helix. Figured out the dimensions, how big each turn is, how long it is, what the radius is, and the overall architecture of the DNA. So I brought this just to show, actually this is in here, this perfect timing, but this is the DNA, structure of DNA. Little molecules all arranged, all the atoms in space. So the red and the yellow going up along the edge, so like the rungs or the arms of the ladder are all the sugar phosphate backbone. And then in the middle is all the ATCG, all the nitrogenous bases connected to each other. So it spirals all the way up. It's a right-handed, double-stranded helix, DNA helix. So I'll just, if you want, you can pass it around. I mean, it's kind of clunky and heavy, but you can look at it from like table to table. It's pretty neat. So that's what they do in chemistry too. They do all the little balls and put them together so they can learn the overall structure. I will say Rosalind Franklin spent her life doing x-rays and doing x-ray diffraction and studying DNA and studying cancer and studying radiation. Ultimately, she died of cancer at the age of like 35. So this is before we knew how carcinogenic a lot of different things like x-ray exposure and radon and some of these different things were. And she handled all of that stuff and did it a lot. So she ultimately got cancer. I believe it was ovarian cancer and died very young. So there's been books written about this now too, but in a lot of our science books before, Rosalind Franklin was barely mentioned. Like, she's not even there. It's James Watson and Crick, right? It's not her. So I like to give tribute to her, who actually, the grad student, who in the old white guys' labs, right, that had the money and stuff, she's the one that actually did the photos and did the pictures that actually resolved the structure of DNA. More structure of DNA, just showing how they pair. A goes with T, C goes with G. The little dotted lines represent hydrogen bonds. So hydrogen bonds, again, between the nitrogen or oxygen and hydrogens. When they're adjacent to each other, they get to make those hydrogen bonds like we studied and we looked at in water. The same thing, there's hydrogen bonds among those bases. So the rungs of the ladder are the bases, and the side or the arms of the ladder are the sugar phosphate backbone. Okay, the code of DNA is in here. This is what matters. And notice, hydrogen bonds, are they weak or strong? Sorry, I know it's kind of clunky. They're weak, right? So we can rip these apart. Enzymes can actually tear the DNA apart like this, unzip it, get enzymes in there, and replicate it, duplicate it, so you can double up your DNA. That's before a cell divides, it will do that, before it undergoes mitosis, okay? You can also do that when you're expressing different genes. So unwrap, copy, and then go on from that point forward. So these can be ripped apart and then zipped back together. So unzipped and then zipped again. So DNA is really the blueprint for you, okay? So all the genes, the units of inheritance, are coded for by your genes. Is there a question?
Yes. In the previous picture, the A was always bigger than the T, like in the visual.
Oh yeah, so something you're noticing, and you don't have to know this, in Bio 1 you'd have to know this. Adenine and guanine are purines, they're double-ringed nitrogenous base. They have 2 rings, so they're fatter. Cytosine, thymine, and uracil are pyrimidines, and they're single-ringed, one cyclic ring, and so they're smaller. And that's how people learned, they figured out how they paired together. A 2-ring always goes with a 1-ring. So a purine always bonds with a pyrimidine. That's the way they set up. And that's why they go A with T and C with G. That's how they pair. So good observation, yeah. I was gonna have you learn all the tiny little nooks and crannies because it gets a little bit involved, but yeah, good eye. All right, so genes coded for by the DNA. They make you, okay? Every single protein, part, structure in your body is coming from your DNA. It's coming from your blueprint, the code, okay? So the genes code for distinct proteins. Your body, your DNA does not make sugar and it does not make fats. We have to take those things in. But it can make some protein, and it makes a lot of protein. All the building blocks of your body, the connective tissues, the muscle, the structures, the bone, all that is made with the proper nutrients, of course, the proper vitamins and nutrients. But those are all made of proteins. Each gene is a distinct sequence of nucleotides and their bases. So that A, T, C, G, the sequence of how those occur is what determines each individual gene. They can be hundreds or thousands of bases long. So one gene can be a series of A, T, C, G, C, G, 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 C, C, A, 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 T, T, right? However many times, hundreds to thousands of times that will represent one gene. Now, interestingly enough, I've studied a lot of genetics and the human genome, specifically looking at fertility in women and how that correlates to certain genes across the genome. And we know that about 2 to 3% of the entire genome, all the DNA, complexed into all the chromosomes. We have 23 pairs, or 46 chromosomes. Only about 2 to 3% of the DNA is genes. The rest is other stuff. And we are still trying to figure out what a lot of that other stuff does. A lot of it's regulatory, or it's junk DNA. Not really junk, there's other things that it does. But for the actual coding part of the DNA, it's only about 2 to 3% of it that is actual genes. So there's a ton of material, ton of information in your DNA. Lots of research. Now we know the entire sequence of bases of the human DNA. And this was a project of many, many years called the Human Genome Project. Many different scientists and labs and researchers from around the world working on this Human Genome Project to be able to sequence and know every single probable base at all those locations across the genome. Of course, there's variability among people, right? But the whole overall human genome was sequenced. Now we went on to sequence like fly and chicken and cow and cat, and even things getting into other types of creatures like invertebrates, like different kinds of marine animals. They're starting to get the genome sequence of all kinds of different things. And you think, well, yeah, that's cool, whatever, doesn't really matter. This allows us to figure out so much about who we are or who that structure or organism is, because you can figure out what does the code say, what proteins do those code for, what are those genes, how does this affect disease and illness and all these other, the physiology of the body, if we can figure out what the actual blueprint is, what those genes are and what they code for. So there's enough letters, just to be a dramatic effect, there's enough letters, the nitrogenous bases that are the code, to fill every single book in 3 huge libraries. Every single book along every single row, just filling up all those entire books, all of them. That's just the letters of the nitrogenous bases in your DNA. So a lot of improvements because of knowing the human genome sequence overall, the genome that we have comprised as human beings. Now we have improvements in genetic counseling, genetic diseases, gene therapies, medicine used to treat lots of different illnesses. We're starting to learn a lot more about sequence variations. So at particular places in the genome where there's variations, normal variations, I might have an A, you might have a G. Those little variations can predispose you towards certain illnesses, certain diseases. The effects of metabolizing different drugs might be different in me than in someone else. We would not know this if we didn't study the genome and genetic variation among people. And things that you're able to digest or not, or what you're allergic to. A lot of those come down to genetic variations or genetic, I don't want to say mutations because they're not always mutations. They're just variants. Some people are different, everybody's different from everybody else, right? So we all have some of those variabilities. DNA gets all wrapped around proteins, packed up and coiled real tight. It gets into its superstructure. That's just the basis, the molecular structure of DNA polymer. It gets wrapped around and complex and coiled and packed into chromosomes. That's an electron micrograph of an actual chromosome. So they look like little X's. It's actually a duplicated chromosome. So here is one complete chromosome or chromatid and another one. So they're actually sort of duplicated and they're connected right at the middle. So the genes are actually, the DNA is actually duplicated. And in this case, the cell gets in this chromosome, the DNA gets packed into this chromosome state when it's going to undergo division. So when the cell is going to divide, it packs into these chromosomes. Like when you're going on a trip, what do? You're about to go on a trip, you're going to go away for a weekend trip. What's the first thing you do?
You pack.
You pack, okay? Do you just throw a bunch of crap in a bag all anyway? I mean, maybe you do. No offense, you do. Sometimes if I'm in a hurry, hey, just ball it up, go. But in general, my daughter's a pro at this. She will fold and roll everything. All her little chonies are just tight, little, rolled up in her bag. She can fit like 80 things in one little bag. That's what your cells do. They need to wrap up the DNA and scrunch them up into little packages because you're going to be going on a trip. They're going to be dividing and getting separated and going to 2 separate cells. So this is in this final superstructure where they're packed into chromosomes because they're going to be moving. They're going to be doing something. We're going to talk about that soon. RNA, how are we? Do we have a couple of minutes? We have 1 minute. Okay, so read RNA on your own. I'll come back to this. At the very end, I just talked about the function of RNA. I'll come back to this on Monday. And ATP. Oh, I guess that was it, RNA and ATP. Okay, so look over that. I'll come back to that on Monday. If you didn't do your application of the protein, just bring that on Monday. Do that as homework and bring it on Monday. And I would say for the schedule, I'm going to post on Moodle, and then I'm going to bring you copies of just our tweaked schedule, our edited schedule. And then someone asked about the reading. If you're keeping up with like the reading of where we're at, or you're doing the reading of what we're just about to go into, you're perfect. Because we're a little behind in the reading. So if you're way up at mitosis right now, you're like way ahead. Which is fine, you can do that. But I just like to have you do the reading like right before we do the lectures on those materials, right? So, all good.